Plate surface coating is typically recommended for non-adherent cells to promote even distribution within the well. Most adherent cells can be directly added to the wells as they will attach to the surface without the need for additional coating. Most reagents require sitting in the well for a certain amount of time, being aspirated out of the wells, and then being allowed to air dry until all liquid residue is gone. Invert your tube of cell suspension several times to gently mix and make sure the cells are evenly dispersed before pouring into your reservoir. Reverse pipetting is a technique to dispense liquids while minimizing bubbles or splashing. Aspiration of the liquid is started from a fully depressed plunger, also known as the second stop. As you slowly release the plunger to aspirate the liquid, excess volume is drawn into the pipette tip. Dispense the desired amount of liquid by pressing only to the first stop, leaving a residual volume in the tip, which can be discarded. After seating cells, let the plate sit at room temperature on a flat, level surface for 15 to 30 minutes for adherent cells, or 30 to 45 minutes for non-adherent cells. This allows the cells to begin to attach to the plate and ensures even settling and distribution of cells across the well for optimal data collection inside the instrument. With proper reverse pipetting, bubble formation should be minimal. Any remaining bubbles can be removed by gently squeezing a wash bottle containing 70 to 100% ethanol with the inner straw removed over the surface of the well. Pre-warming culture media and other reagents to be added to cells and allowing the plate to warm to incubator temperature for 30 minutes before initiating scanning will minimize condensation, which can obscure images.